Hi, my name is Kevin. I'm a postdoctoral research fellow at the University of California, San Diego. And I've been working on aging, and I'm really honored and humbled to receive the poster award. And I want to share some of my research about how aging may contribute to ALS and how we can learn from some of the research that I've been doing. So, aging is a major risk factor for ALS. So, when we look at incidence of ALS within people, we see that it's correlated with age. There's a steady increase of ALS cases as a function of age until you hit about the 80s, and then it drops a little bit from there. Um, Aging is also a known risk factor for other neurodegenerative diseases such as Alzheimer's and Parkinson's. And so in general, this is something that we should be focusing on in our research, but it's one of the most difficult problems for us to address as researchers. This is because we tend to rely on this system on the bottom here, in which we are trying to generate neurons, and we do this by making these things called stem cells. But the issue here is that this kind of makes all of our cells look young in the test tube. And so that's an issue. We want our cells to be old, like what's happening in people. But this is kind of a difficult problem for us to get around. If we want to try and age these young-looking cells in our petri dishes, we have to wait 40 years, and nobody wants to wait 40 years for important results in this field. So to get around that, we've used this system on the top of this diagram in which we're taking cells that were generously donated by patients, and we are directly converting them into neurons. And this allows us to study these neurons in the context of aging, because all of these aging markers that the person accumulated over their lifetime are retained by this differentiation process that we're using. And so this approach allowed us to look at a lot of different changes within neurons as a function of aging. And here I'm just summarizing the main results from three different components that we looked at. In general, we saw that there was very widespread changes to RNA metabolism. And so this encompasses all sorts of RNA-related processes, the production of RNA, the transport of RNA, the types of proteins that bind to RNA, and how RNA is stored and then eventually destroyed. These are all really relevant to ALS phenotypes because the main proteins that are implicated in ALS are all RNA binding proteins. They'll interact with RNA. And so in our results, we found that there was three different things that we focused on that were really interesting. One is that first we found that a lot of RNA binding proteins, including the ALS linked protein T43, are really mislocalized as we age and have older neurons. You can see that here on the left, in which this TDP43 protein is mostly nuclear, or it's contained within the nucleus of cells in younger cells, but then it translocates to the cytoplasm of these neurons as in the older cells. We also notice really marked changes in the splicing. So this is the part of the production of RNA, and it changes like which instructions are encoded onto other RNAs. So TDP43, which is the protein here, is known to help with splicing. And we see that in these older neurons that have the changes in TDP43 localization, there's also changes at TDP43 binding sites in terms of the production of the RNAs. For instance, this RNA here that I'm showing has two exons, or kind of instruction regions, that are included in young neurons but are excluded in old neurons. And so this can change the functions of the proteins that are encoded within the RNA, and can also change their localization and all sorts of things. And this is known to be important in ALS as well. The last thing that we noticed was that there was the chronic accumulation of RNA binding proteins into these things called stress granules. So stress granules are thought to kind of help your cells respond to stressors and make them more resilient. And what we noticed is that these older cells were already forming these bright puncta called stress granules, even when we didn't do anything to them, to the cells. And so this seems to imply that the cells are already stressed to begin with. And actually, the chronic sources of stress can be bad in that your cell can't respond to new stresses very well. And so together, the data that I'm showing here, and it's just a very brief overview, we think that aging is one of these cumulative risk factors that contribute to ALS pathology. And this will better inform us on how to develop treatments that are more tailored to ALS in the context of aging, now that we are aware that this is such a prominent risk factor and can kind of destabilize our neurons. And so already in my preliminary results, we've seen that we can treat with this inhibitor that was already developed and reduce the number of stress granules that are forming within these older neurons. And of course, there are other potential treatments. I'm not very good, or I'm not well versed in drug discovery, but we think that if we applied this platform to look at or to try and look for new drugs that might help in the context of aging and ALS, we could find things that are helpful treatments and are helping to reverse some of these aging related defects. So with that, I really want to thank you for your time, and I'm happy to take any other questions you might have.